So the problem with recording is that, you know, any idiot that I make of myself is now on the internet in perpetuity.
Immortalized, yeah.
Yes, exactly. There's a terrible, I did a synthetic biology workshop years ago when I started as a PhD student with Victor De Lorenzo. And we had to make these videos which were stuck on YouTube afterwards, and it's so embarrassing to look back. Really terrible.
But you know what, you know, your work will be available freely to the community.
This is the benefit.
This one. That's actually very, very great. So you are thinking about faculty, you know, position, and this one will be seen by your potential recruiter. And then you are selected out of, you know, more than 100 different people, and that means, I mean, you are rising star. So I mean, that means, I mean, the recruiter will look at, you know, your CV for sure, and in this video as well. So I'm not giving you pressure. But
No pressure, but everything hinges on this video.
So how's the weather in London right now?
You know, it's not bad. It's a clear, sunny day.
Oh,
Yeah.
Actually, the sun looks like it's just starting to go down, so you're going to get some quite interesting sunset scenes in the background of our talk.
ah, that's nice. I mean, I always kind of imagine London is something like foggy and, you know, a little bit cold. I mean, in the winter, that's kind of my impression, but that's not gonna.
It's quite a nice, like, crisp, cold day today, but
Yeah.
there's sun out.
Ah, I see. I mean,
No
you know
fog
what?
anymore.
You know, my area, you know, used to be, you know, very cold. I mean, below, you know, freezing temperature. But even today, I mean, we have now I'm seeing the 42 in my computer in the screen, and then it'll be probably 50, you know, noon. So I mean, you know, I'm kind of.
I don't work in Fahrenheit, so I'm not entirely sure what that means.
I am nodding slightly going, that sounds mild, I think.
42 is, I mean, Celsius wise, I believe, let me see, probably 6 to 8 Celsius. I mean, and then, or something like that. More than 5, I mean, below, yeah. So something like that. So I'm, you know, I used to be, you know, Celsius, but now, you know, Fahrenheit because here.
Yeah. S being the oddity of the.
Okay, so let me see. Okay, so I guess, I mean, if we probably want to start now. Okay, so good morning, afternoon, evening, hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you for joining us, the 16th seminar and Nature Communications Day. Interestingly, today is also Make Your Dreams Come True Day, and this unofficial sort of holiday encourages people to try to achieve their dreams. In fact, of course, every day is holiday, an important day to me since I miraculously recovered from many years of literally dying. And now I enjoy every single second of my life. And of course, this seminar series is my favorite. And then this is always kind of, you know, the Thursday is always kind of my favorite day now. And if your New Year resolution is to publish great work, you can consider, of course, always nature journals, including Nature Communications and Nature ChemBio, etc. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Ross Cloney. I would not need any introduction of him, but he is a powerful man, of course, senior editor of Nature Communication, especially his support synthetic biology tremendously. And I met him multiple times in synthetic biology conferences and one of the IGM seminars. And that clearly indicate he cared about synthetic biology and he is big supporter of synthetic biology. So I'm very, very thankful him about his support. And what else do you want to know about him? So let's submit our great paper to him. And Ross, the virtual floor is all yours now.
Thank you for that introduction. So I'm not going to take up tons of time because I want to give Alex the full opportunity to talk about his fantastic work. But I want to start by thanking Professor Moon for inviting me to give a short talk today. It's an absolute great honor of mine to be involved in this seminar series. And more importantly, I want to thank you and thank everyone involved in this series for all the work you've done in organizing it and hosting these seminars, particularly because these seminars have an absolute fantastic wealth of knowledge of the research they represent, and the really strong focus on early career researchers. Even in my relative short time being involved in synthetic biology, it's advanced so far and so quickly. And I'm just really excited to see what the next generation of researchers who, have quote unquote grown up in the synthetic biology community and the ecosystem and the ethos of the synthetic biology community from the very start of their academic lives. So I think you covered who I am, but for anyone who doesn't know, I am an editor at Nature Communications. Most of my time with the journal has been spent handling their synthetic biology and their genome engineering content. And over that time, it has been my absolute privilege to handle this content and to be warmly welcomed by this community. In the last few months, I've also recently become head of a team of editors at the journal. And my team handles the full breadth and depth of biotechnology and the applications of biotechnology to the world around us. And I constantly find one of the absolute great joys I have of being an editor is to see how a field is developing, how it is forging these really interesting and fascinating interactions and new connections with other areas of scientific research, and to see how it's moving out of the lab and into day-to-day life. And this is particularly an area where I'm fascinated and really interested and really excited to see where synthetic biology is going to be heading. One of the aspects of synthetic biology that I've always admired is the ethos it has of openness, of engagement, and of bringing in thoughts from beyond the lab bench to ask questions that are going to be absolutely vital as it moves from something that's done in a sort of an academic setting to something where, I hope one day you can pop down to your local shop and pick up a synthetic biology product, item, something that has been designed, built, tested, learned, and brought from the proof of concept setting into the that's $2.99, I'll pick up a dozen of them. And one of the reasons why I find this transition into the everyday really fascinating is, and I really don't want to dwell tons in the last 2 years, but I think we've had a very powerful demonstration of the impact biology can have, and also the ability of biotechnology to confront and address some of the really important questions the world faces. And I was thinking about this a bit the other day, and I find it fascinating, and kind of like fascinating now how mundane I find it, that at this very moment, in my flat, I have a box of 7 lateral flow antigen tests for COVID, 7 relatively cheap and easy to use pieces of biotechnology that allow me to know something about my own personal health and to help inform the decisions for myself and the people around me. And these are really rudimentary demonstrations of biotechnology compared to the advances that synthetic biology will be bringing over the next few years or so. And I say this a lot whenever I'm asked to give talks or whenever anyone lets me talk to them about synthetic biology. But I do genuinely believe it's going to be a very transformative discipline, something that's going to have potentially very radical changes for the world around us. And that it is important that it keeps reaching out to other fields, to other stakeholders in its development, and to have these really open and honest conversations about the advances it makes possible and the technologies that can be developed using synthetic biology, how they will be used, and to ensure that the people impacted by them are involved and empowered in the discussions around their use. Because I think, it's not hyperbole to say that synthetic biology has the potential to impact so much of the world around us, from our personal health to how we manufacture products sustainably, to how we manage, adapt to, even engineer ecosystems in the face of climate change. It's been a joy to see the field develop and to see how seriously it takes the questions about what changes it enables. And, seeing all this as an editor, questions occasionally pop into my mind, which I'm going to rattle off a couple of them now. I take no credit for these being original questions. I know they are very active areas of discussion within the field at the moment. But I think they're ones where the answers are going to be really interesting to how everything develops. For example, things I occasionally think about, and Alex might be able to help on the first question. What does it look like in terms of our personal health when we can engineer our gut microbiome? What does it mean that if engineered microbes can produce sort of anything carbon-based, what impact does it have on traditional manufacturers or farmers or local producers? A question that I find absolutely really fascinating. If we can produce things using microbes and everyone could produce these high-end products on their kitchen countertop with a sort of sleek-looking Nespresso-like fermenter, what does that mean? As we develop more powerful technologies, things around engineering complex microbial consortia, engineering gene drives, can we or should we engineer ecosystems for our benefit? Should we do it to address some of the ecological damage that we've caused? Or should we be like, this is something that, perhaps we shouldn't even consider engaging in because of the complexity of the question? And when I say should we or what we should do, when we talk about we in terms of using these technologies, what groups are being included in the discussion and what groups are not being included in the discussion, and how do we bring them in? What can be done to ensure fair and equitable use and benefit of these potentially very powerful technologies? And one of the questions that I actually think about quite a lot is how does the synthetic biology field respond if there is a problem and we present a perfectly viable, useful synthetic biology approach to solving it? And the answer we're told is, no, thanks. It's not for us. We don't want to address the problem in this manner. It's a bit of a cop-out, but I'm not going to give any answers to any of the questions I just posed. But ultimately, I think I wonder a lot about what does everyday synthetic biology end up looking like? And particularly, what does it look like in places that are not currently, the centers of synthetic biology research? What does it look like in places that aren't California or London or Singapore? So with that, lots of questions and not a single answer for any of them. I am more than happy to turn the remainder of this time over to Alex, because I want to hear about the research that will be involved in both posing and hopefully answering some of the questions I just put forward. Thank you for your time.
That's amazing. Thank you so much. Amazing insights and inspiring talks. Yes, we as researchers should address global health, climate crisis, and food and sustainability issues. And I believe Alex will address some, even though not all of that. And also by accepting and also sadly rejecting our papers, you guide us, also define the direction of SynBio and biotech into the future. So I'm really thank you for being a pioneer for all of us. So thank you so much.
Thank you again for inviting me. I think mostly I am thankful to be part of the conversation the field is having.
Sure. I mean, you should be part of the conversation. Otherwise, this would be a monologue, not the conversation, right? So thank you so much again. So now, okay. Now, of course, the main speaker of today with a longer introduction. And in fact, both speakers today from London. So I could say, I mean, today is also London Day. So Alex Fedorec is a postdoctoral research associate in the Division of Biosciences at University College London. His broad research interests cover two areas: microbial communities and biological information processing. Both of them I really like it, and which he hopes to bring together as microbiome computing. He has a background in computer science, having completed his bachelor degree in that subject and worked for a short period as a software engineer for BlackBerry. Probably young people even do not know what BlackBerry is, but this is one of the, you can say there's cell phone with some, messaging, kind of function, I guess. He joined Professor Chris Barnes' group at UCL in 2014 for his PhD and has remained there ever since to continue his postdoctoral research. He also co-founded and continues to run the London SynBio network, which brings together researchers at the university and institute spread across London. This provides an inclusive forum to share ideas and start collaboration with a particular focus on engaging young and underrepresented researchers. I think highly of him, especially because of the London SynBio network activity to provide a platform for young and underrepresented researchers, which is the same motivation and spirit of SynBYSS, this seminar series. So thank you so much for that activity and then your wonderful research activity as well. So Alex, take it away. It's all yours now.
Thank you so much. I'm going to just try and share my screen for you. See and hear me okay?
Yes.
Yeah. Wonderful. I really want to start by saying a massive thank you to TSOC for putting on this series. He's put in a huge amount of work to provide this platform for people like me. And I'm incredibly flattered to be on the bill with, you know, some really, really amazing researchers who have already given talks last year and continue to give talks following year. So with that, I also want to slightly apologize because I've developed a little bit of a tickle in the throat in the last couple of hours. So I've got a glass of water. I apologize if I start coughing. Okay, so today I really want to talk to you about kind of one of the aspects of my research, and this has been research that I've done kind of in my PhD years and continue to do in postdoc years. And that's around engineering and controlling synthetic microbial communities. Along the way, I'm going to highlight work done by a lot of really smart and really driven people. And I've got their photos up on my slides. So I encourage you to look them up and look them up. I wouldn't have been able to do most of this work without help. So to start with, I want to say, like, the progress that we've made over, I mean, synthetic biology is in theory 20 years old, but you know, there's been developments in the decades beforehand. But even in that time, we've come to the point where we can get cells, engineer cells, to do some truly incredible things. And I think this example that I've got up here, the cello system from Chris Voigt's group, is really the epitome of that, where we can go from coding up a logic function in a computing language, and it will translate it into a sequence of DNA, which you can transform into cells, and it will reliably and reproducibly perform that computation. And I think if anyone isn't amazed by these kind of strides that we've made in the last few decades, I don't understand them. However, one of the struggles that we're starting to come up against and has been recognized for the last decade is that there's only so much that we can ask a single cell to do. There's a limit to the number of parts, proteins that we can express in a cell. There's a limit even if we start moving towards RNA kind of regulation using CRISPR-Cas technologies. There's a limit to how many of those we can express in a cell due to orthogonality, due to burden, due to a whole host of issues. And so it's been suggested many, many times over that we should be starting to think about how we can engineer communities of cells rather than just monocultures to perform ever more complex functions. And this review paper from a few years ago, I think this figure really kind of encapsulates all of the benefits of starting to think about things in a community sense. It allows us to bring in the kind of the real benefits of the engineering disciplines from kind of modularity and compartmentalization. And it also allows us to expand the kind of things that we can do with existing biological systems, taking advantage of those existing biological systems. I think probably more importantly than the ability to build ever more complex things is that even the simple things that we're going to build, the applications for them are going to be in environments where there's already microbiomes present. So whether we want to introduce therapeutics into the gut or to start doing bioremediation of contaminated ground or climate change mitigation strategies, all of these environments, there's already going to be a microbiome present. And therefore, anything that we introduce into these environments is going to have to interact in these environments. And if we want to do this, you know, ethically, then we need to be able to predict how these things are going to interact and try and predict the outcomes of these interventions. Okay, and so my research over the last few years, as I said, has been really focused on how we can control synthetic microbial communities to start with, and then how we can start to interact with existing microbial communities. And so here, so I'm just going to bring up a laser pointer. So here we've got an example of an environment and in that environment, we've got 3 different species of microbe. And these microbes will interact with each other through molecules that they secrete into the environment and which they consume from the environment. There's also going to be a flow into and out of this environment, this particular environment, of a variety of different molecules, whatever level you want to think of these things at. So in this situation, you've got interactions between the community mediated by molecules, and then you've got environmental effects to think about. And so I want to talk to you today about 3 different stories, 3 different kind of pieces of work which we've been focusing on. The first is going to focus on situations in which we've got control of the environment and also potentially control of the interactions which are going on in the environment. The second story is going to be talking to you about what happens if we don't have control over the environment. So we're going into a situation where we don't know what's coming in and out. However, we do have control over the community which is placed into that environment. And finally, I want to talk to you about possibly the most kind of significant challenge to me and the application with probably the most potential is the situation in which there's already kind of this existing microbiome. It's in an environment which we don't have control over, but we want to introduce something into that environment which will perform a task or is able to manipulate the environment, manipulate the community in a particular way. And so the first thing that I'm going to talk to you about is this situation where we've got control over, in theory, control over everything. And the way that we approach this is using reinforcement learning. And this was some really, really great work that Nathan did during his, well, he actually started this as a master's student and continued it his PhD. But this is using reinforcement learning in order to try and control how these communities interact. And for those of you who are unfamiliar, reinforcement learning is about training a system in order to learn what actions to take in order to push the environment towards a goal, right? And so in this case, the agent is able to take actions which influence the environment and the community within the environment. And then from that, it receives a reward as whether it's pushing that environment towards a state that is that is part of the goal. And so the agent measures the state of the environment and decides to take an action based on the state and its expected reward. And so here we use kind of quite a simple system in which we have 2 microbes which are competing with each other. And these 2 microbes are they require particular molecules in the environment. So they're both competing over this blue molecule, which could be a carbon source, for example. But then each of them requires a particular nutrient. So this would likely be an amino acid that each of these strains are specific for. And so there's competition involved, but there's also an extra nutrient. And the agent in this situation is able to choose which of these nutrients to add into the environment. And its goal is to try and produce a community with each of the 2 strains at a target density. So what happens is the agent will measure the kind of state of the environment. It will take an action, which will be the addition of nutrients. It will push the environment and the community to a new state, and it will continue to take actions. And so we use this method, which sounds a little bit fancy. It's called neural fitted Q-learning. But effectively what that means is that the agent starts off with no knowledge of what its actions are going to do to the system. And so it just picks randomly what nutrients to add into the environment. But from these kind of random choices, it starts to learn how those actions are going to affect the community. And then through multiple steps of training, here we've trained it over 30 different what we call episodes, we start to reduce the amount where it just chooses kind of random actions. And instead it begins to rely on its own knowledge that it's developed of the system. So that's that blue line. We're reducing the kind of random explore rate. And as we reduce that and it starts to rely on its own knowledge, you can see this black line is increasing the amount of reward that it's able to achieve. And what we finally achieve is these two communities. So this is over time, and these are kind of population levels of the two communities. We're able to stabilize two communities around target densities. And you can see that these communities are actually oscillating around these target densities, and that's because the agent is only able to kind of add, high concentration or nothing, rather than kind of fine tune the concentrations of anything. So it's what's called bang bang control. What the agent is actually learning is some idea of what's called the value function. So it learns where it's going to get the best reward, and so it's optimizing its own actions in order to try and get the most reward that it possibly can. And what the outcome of this is what's called a policy. And in this case, because this is quite a simple system, the policy is quite easy for us to understand. And here, sorry, the text is really small, but along the x-axis, we've got the population of the kind of blue population, so N1 cells, and across the y-axis, N2 cells. And each of these colored patches are the action that it's learned to take when the population is at a particular level. So here, when we've got lots of N1 cells, but not very many N2 cells, it decides to add the amino acid, which increases the population of N2 cells, but doesn't help out the N1. So this is feedback going on. So this allows us to, I think, T-Stock, you're unmuted. Thanks. So this gives us kind of a very nice interpretable understanding of what the agent has learned to do. What's really cool is that actually we can learn some reasonably complex behaviors, which would be quite hard to reason about yourselves. So in this situation, the setup is quite similar in that there's kind of competition over one nutrient, and then each of the other nutrients is unique to each of the strains. But in this situation, each of the strains is producing a molecule which is then used in a downstream process to create some kind of product. And here we're learning to optimize for the product outcome rather than learning to balance the community. And it learns that to optimize the product, it needs a particular kind of ratio of the community, but that's not fed into it. And what's really unique and useful about reinforcement learning is that we don't actually need any understanding of the mechanism of the interactions between the strains. So it's a model-free approach to control. And that means that we can control some reasonably complex systems as long as we have enough kind of time to learn. Now, where I really want to get to with this research is to be able to control systems dynamically. So in my ideal world, I would be able to play computer games with bacteria. So here, for example, we've got a situation where we have one of the populations at a very high level or one of the populations at a very low level, and we could feed, dynamically control, guide our community through different states, depending on what's required at that particular time in a process. So for example, if I don't know if anyone's like interested in gardening at all, but if you have a compost heap in your back garden, then you might know that through different phases of producing compost, you get different communities developing in different temperatures. And so there may be processes that are important in the future where we need to be able to guide the community through these stages. Okay, so that's reinforcement learning, and that's if we've got kind of control over the environment. What if we don't have control over the environment? So in this situation, we need to be able to develop a community which has the ability to self-control, self-organize. And there's been quite a lot of work done on this in the past, predominantly in the kind of the bioindustry world, producing various molecules using very simple communities. And the approach there has predominantly been using cross-feeding. So in this situation, this bacterial strain is producing a molecule which is required by the other strain in order to grow, and vice versa. Which is required by the other strain in order to grow. And so there's this interdependence built into this system. And so that's been the predominant kind of paradigm for bioindustry. But I think in synthetic biology, it seems to be the researchers are slightly more sadistic. And so there's been a lot of papers which have involved getting bacteria to kill themselves rather than to help each other out. And so if we look at this one, in this system, we have a strain of bacteria which is producing a toxin within itself, within the cell, which kills itself. And the production of that toxin is regulated through the production of a signaling molecule. And so that cell produces its own signaling molecule. And when the density of that molecule is high enough, it turns on the production of the toxin and the population kills itself. And so this is the kind of, in synthetic biology, quite a famous system of a synchronized lysis circuit from Jeff Hasty's group. And they stuck two of these things in the same environment and managed to get some semblance of coexistence. And these things have been kind of linked up in more interesting ways. So you get kind of cross-dependence of strains and combinations of these things. So yes, so there's been a lot of work on how you get bacteria to kill themselves reliably. The problem with getting things to kill themselves is that there's a lot of mutational pressure in order to escape from these systems. So as soon as you get a single point mutation which inactivates that toxin or stops the production of it, the population will quickly run out of control and basically break your system. So these systems tend not to function for a very long period of time, though there is also a lot of work being done on mutational stability. So going back a little bit in time, actually, we were looking at ways in which we could control something called plasmid stability. And it turns out that these problems of self-control of a population, of a community, and plasmid stability seem to be interrelated, or at least they are in my head, because plasmid stability is inherently a problem of competition. For those unfamiliar, what plasmid stability is when we have a genetic circuit carried on some plasmids within a cell, there's a probability that at division, all of those plasmids are going to be in one half of the cell, and you produce these plasmid-free daughter cells. Now, the problem is that the genetic material carried on those plasmids almost always produces a burden to the cells that are carrying it. And so these plasmid-free daughter cells very quickly take over the community. And so we had been doing some work with Tanel, who was a PhD student in the group, and Anjali, who was a student in Taudanino's group, where they were working on cancer therapeutics. And we had been looking at kind of the traditional method of dealing with plasmid stability, which is these toxin, antitoxin, or addiction systems. And they work by basically producing an intracellular toxin, so a toxin inside the cell, and the antidote to it. And the antitoxin degrades faster than the toxin. So if you lose the plasmids, you very quickly get kind of a concentration of toxin, which is enough to kill the cell. The problem is that if you don't have enough of that toxin within your cell at division, when a plasmid-free cell arises, then you have this problem of escape again, and very quickly your engineered population. And so I had read about these things called bacteriocins, which are toxins which are produced by the bacteria and then secreted into the environment rather than held within the cell. And they would then go off to kill anything which isn't, which is susceptible to the bacteriocins. And in this way, I thought we could effectively produce like a cloud of killing. So you would be policing the arising of plasmid-free cells rather than expecting them to kill themselves. And what we showed was that in this work was that the bacteriocins were better than the kind of the standard toxin antitoxin system, which was used in synthetic biology. But we did discover this one toxin antitoxin system called AXTAX, which proved almost entirely infallible. We never got any bacteria to shake these plasmids. So if anyone is interested in situations in which you can't use antibiotic selection to keep your genetic circuits around, you can look up our work, get our plasmids off AdGene for this AXTAX system. It seems to be really useful. Anyway, so now we've got this bacteriocin system which allows us to kill competing cells rather than relying on having to kill ourselves. What we didn't know was whether we could actually control it in any way, and particularly if we could control it in a dose-dependent way. So we just built this very basic system where one of the cells produces the bacteriocin, and we can turn off the production by the addition of a molecule. This data just shows kind of the hazy bit is kind of a lawn of bacteria, and onto that we've dropped this supernatant from this engineered strain, grown in different concentrations of this repressing molecule, and it shows that we can indeed kind of undo the repression and therefore start killing at higher concentrations. So we've got a system where we can now control killing of a competitor. We can control it at distance. What's really useful is that the molecule that we used here is a signaling molecule which these cells can themselves produce. So these cells can now regulate the production of their own bacteriocin. So we've now got a whole bunch of parts which we can start to stick together. We've got strains of bacteria, we've got signaling molecules which we can use to control the expression of the bacteriocin itself. And there are dozens of different ways that we can link these things together. So here we've got these normal arrowheads signify positive interaction. So this strain is producing this signal. A flat-headed arrow here signifies a negative interaction. So bacteriocin is repressing or killing this strain of bacteria. And these ball-headed arrows signify that they could be a positive or negative interaction. So the signaling molecule is able to turn on or turn off the production of the bacteriocin. And so we could set up this system in a huge number of different ways. And all of these interactions can also have different strengths. So it's not just like a huge number of topologies, network topologies, but also a huge number of weights to each of these interactions. And so how do we pick out the best possible system to build from all of these possibilities? And so this is some work that Bez did during his PhD, which was developing this framework using what's called approximate Bayesian computation to search through this vast space and identify things to build which are most likely to be able to achieve our particular goals. And so in this particular example, what we do is we set our desired behavior, which here is kind of stable coexistence of bacterial strains. We need some kind of prior knowledge about the possible strength of the interactions. So we need to put on biologically reasonable limits to these things. And then the software basically samples this vast space and narrows down which systems we should be thinking about building and which are the most likely to be able to produce the interaction, the goal that we are targeting. Now, what's interesting is that we can start to compare how well each of these systems are likely to achieve this goal against each other, depending on kind of different motifs which are in the network that are built. And so what we did was search through this vast model space and perform some clustering. And what you see is that the models that perform best are actually highly clustered around particular regions. And if we pick out what that clustering really means, is what we see is that all of the best performing systems, and these are best performing at different levels of complexity. So from the kind of simplest systems to the most complex systems, all of them have this motif of the cell producing the bactericin, which kills itself. So there's this self-limiting cycle. And then that self-killing is repressed by the competing strain. So we found that really interesting and really interested in whether these patterns are seen in other ecological systems, or whether it's really a kind of a unique motif to designer communities rather than real wild communities. So this gives us some kind of idea of what we should be building, and it also gives us some kind of idea of how complex a system we should think about building. Because you can see here that if we step up in complexity one level, then we get this big increase in the probability that the system is going to be able to achieve what we want it to. But the next step up in complexity, there's a diminishing return. So we can use this to inform, is it worth really going about building these more complex systems? We also did this for even more complicated ones, so 3 species systems, and these are really difficult to postulate about using kind of traditional methods for doing this kind of work. And we see exactly the same kind of motifs present, these self-limiting motifs. And we've recently started looking at, so what happens if you choose poorly? So in this situation, this system is able to achieve kind of stable community existence. But if you get your parameters slightly wrong, so in this case, we're looking at the rate at which this signaling molecule is being produced and the rate at which the environment is being kind of refreshed and diluted. We move from this dark area here where we get stable coexistence through some oscillating states in this light green. And in this red area, we get chaotic behavior. And chaotic behavior is signified by small changes in initial conditions resulting in very different outcomes. And more interestingly, we can actually move through all of these states in the same system. So here we're slowly changing the rate at which this molecule is being produced, and we move from this chaotic system, pardon me, through an oscillating system to stable coexistence, and similarly for dilution. So this is just kind of a warning that if you're going to think about producing these kind of community interactions, it's worth looking at what are the possible outcomes if you get it slightly wrong. Okay, the final story that I want to talk to you about is this situation where we're introducing something into an environment and expecting it to be able to control an existing community. So the way that I thought about this was, if we need to be able to control an existing community, we need to be able to control it at a distance. So we've got the bacteriocins that I've already talked about, but we also need some leg to be able to close the loop. And what we're using here is competitive exclusion. And competitive exclusion is a property whereby if you've got two things competing in the same niche, one of them is always going to exclude the other, because one of them will have, however minute, there will always be some kind of growth advantage, and you'll get one of them in the environment. And so what this system does is the engineered strain here in orange is outcompeted by the existing competitor strain. And it senses that it's being outcompeted by the production of this signaling molecule. And when it senses that it's being outcompeted, it starts to produce a bacteriocin. Now, bacteriocin kills the competing strain, carves out a niche in the environment, but in which it can start to grow. And then once it's grown, it detects that it's grown to a particular level by again sensing the core molecule, and then turns off the bacteriocin and allows the competitor to grow back again. So we have this closed loop. And this is kind of the slightly more detailed kind of construction of the system. I don't want to go into. But so we had to do a huge amount of testing of this system to really understand how this competition is controlled and behaves in different environments. So this is when the cells are at different densities. You can see the kind of the standard growth curves, and we can pick out each of the individual strains in this community and see how they vary depending on the densities that the culture's at and the concentrations of the core molecule that we can add into this environment. So how well we can turn on and off the bacteriocin. And all of this kind of high throughput data collection wouldn't have been possible if it wasn't for some software that we had developed in the lab. Claire was a student who worked with me and is now doing her PhD. We developed this tool written in R, and it's available on GitHub. But effectively, all it does is it calibrates a plate reader, which is kind of the classic way of collecting data in microbiology, and provides a robust way to normalize your data as well. And once you've got well calibrated and well normalized data, you can get real quantifiable numbers with which you can then parameterize models. And without this data, we wouldn't have been able to properly parameterize our models. We wouldn't have been able to show you in a second. And so this is freely available. It's on GitHub, and I'm really happy to answer any questions and help people use this. And this work is also, heavily standing on the shoulders of a number of people, particularly people like Jacob Beale, who've been pushing kind of calibration. What's also really cool is that this has now been extended by Esther, who's at Imperial College in London, and she's developed this protocol whereby you can now, rather than calibrating your plate reader to something like fluorescein or Texas red, you can now calibrate it to the fluorescent proteins, which you're actually measuring. So you can actually get protein numbers rather than just an equivalent number. So I would urge you to look up her preprint at some point. Okay, so now we've got all of this data, we're able to very robustly parameterize a model, and that allows us to work out exactly the kind of experimental domain where we expect our system to be able to function as we expect. And again, here we've got kind of the dilution rate of the system, the environment, and the production rate of the core molecule, which we're able to tune by the addition of the inducer arabinose in our system. We built in this little tune. And so we did, we kind of did an experiment, which is a crude attempt at producing a chemostat environment, but it allows us to do it in a much more high throughput way. So we can do this 96 wells in a plate, and we regularly dilute it with fresh media. and the amount that we dilute it kind of gives the dilution rate. And what we see here, so on the x-axis here, we've got time, and on the y-axis, we've got the proportion of the orange cell to the blue cell. What we see here is in one of the situations, we have the orange cell winning out. In this middle situation, where we expect coexistence to occur, you get nice coexistence, and that is again tunable by the dilution rate. And then in this final domain, we get the orange cells slowly being removed. So this shows that we can control at a distance a competing species in order to stabilize a community. And what I really like about this is that it allows us to start to think about what could this competitor be? What could this community be? Part of synthetic biology's dream is to be able to take things kind of off the shelf and assemble them. And that should be applied to communities as well as kind of parts within a cell. So the system that we actually built is not the most robust system. It was pretty much the simplest system that I could think of in my head that would work. And that's this system. But we can use the approximate Bayesian computation platform that I mentioned earlier and look at, are there ways that we can improve this system? And it turns out, instead of regulating the production of the bacteriocin, if we regulate the immunity of our engineered cell, this produces a more robust system. And interestingly, that's introducing that self-limiting motif that we saw in the previous work. You know, as you add in kind of more pieces, you get more stability. Okay, so those were the three stories I want to talk to you about. So reinforcement learning to control a population when we've got control of the environment. Ways in which we can design communities in order to produce a particular goal. So we showed stable kind of coexistence, but we could have easily decided to pick communities which oscillate or any kind of target. And then finally, this situation where we've only got kind of control of a subset or a single member of the community, and how we can use that to control the outcomes of the large community. And so with that, I would just like to say thank you. There's been obviously, as I mentioned, like a huge number of people who have helped along the way, funders obviously, and thanks particularly to Chris, who has given me a huge amount of freedom and encouragement to for some of these things. Thank you.
Oh my God, thank you so much for fantastic. Also, I also really apologize for the technical issue from my computer. Echoing. Yeah, I mean, so somehow my Zoom has been so tired because I typically use it minimum 8 hours a day for a couple of years now. I have experienced microphone, video issue previously now, you know, audio issue today. So I already see, let me see, the one question, or actually two questions. First question from Amanan. That question is thanks for sharing your awesome work. My first question is around discussions about plasmid stability to help couple growth and plasmid expression. Would another approach be to knock out an essential gene in the host, but re-express it or co-express it from the plasmid? I guess, I mean, he's talking about plasmid addiction system.
Yeah, that's a great idea, and that's been done in the past. We were interested in things which were solely carried on the plasmid, so that they're easily transportable to kind of other strains of bacteria, rather than requiring you to kind of engineer the genome of your bacteria to knock out, for example, amino acid. But yeah, that as you said, that's definitely a way to do.
Okay, before going, Ross, do you have any comment or question?
No, nothing to say beyond absolutely fantastic talk. And it's great to see how an idea progresses through the stages from the theoretical to the implementation.
I should say a big thank you to Ross, because he was a fantastic editor in publishing some of that work.
Wonderful.
It was fantastic work to handle.
Okay, so next question is John Marken. Wonderful talk, thank you. The question is, in your final project, the unengineered competitor was represented as a single strain, but how do you anticipate that your result will scale if that competitor was instead a community of its own? Interesting question. One could imagine that each strain in that community would have distinct growth rate and susceptibility to bacteriocin, etc.
That's a really good question. So initially, our target would be to start producing multiple bacteriocins, and bacteriocins can be engineered to be pretty specific to particular strains of bacteria by engineering the way that they manage to enter a cell or recognize different cell types. So you can target bacteriocins to specific parts of the community. The question then is, how do you know when to target each of these parts? And so the way that we've designed the system so far, really the kind of controlling strain is just sensing its own population level, and not really understanding how the competitors are getting on. I think at some stage you would have to start trying to work out, is there a marker that you can start sensing in the environment? Is there something that's being produced by one of those other constituents in the community that you can use to control the production of bacteria? I think that would be the simplest way. Simplest is relative, but I think that would be the most sensible way and probably the most robust way. There are probably other ways that I haven't yet thought of that you can do. You can do this tuning just through the engineering, single strain. I think they'll start introducing quite a large reduction in the robustness of the system.
Okay, so let me check whether any other question we have. Let me see. Actually, I have question, but I want to double check first.
Can I also ask a question?
Yeah, go ahead.
So I find it really interesting because, you know, you've got this wonderful way to model different interactions now, and particularly in the work you presented in the latter part of the talk, you can now engineer a single control strain to control a mixed community. What would you see this being used for?
So this work initially started when I was trying to think of ways that we could ensure that bacterial therapeutic that you would introduce into the gut, you could ensure that it would be stable in that environment, and also ensure that it wouldn't just wipe out everything there. So it's, you know, the production of the bacteriocin and control of the community is dampened. And not just that you could predictably know that there is a particular quantity of your engineered strain environment. This is why the kind of tunability was really important. So that's the application that I was initially kind of thinking of the system for. Therapeutic delivered into the gut. It could carve out a kind of a niche for itself. You would know that it's stably there at a particular kind of. The other things that we've been beginning to think about are bioindustrial applications, always going to be interesting and probably in the kind of medium term more amenable to biological engineering. And so situations in which you kind of have specialized strains of anything, which are producing kind of part of a pathway, doing part of a process, where you need to balance those could potentially be useful, particularly with a view to being able to take things off the shelf, which have already been optimized to produce the most of something. You don't then want to have to tune that already optimized strain in order to have to be able to work perfectly with this other strain that you've taken off the shelf. We can have a third strain which is able to do the tuning without having to re-engineer other things. I think that's a nice goal, to be able to really take things off the shelf in biology, I think is one of the goals.
Cool, like a sort of pick and mix approach to a complex microbial consortium. Exactly,
and we're getting there with the genetic parts. We can now take a promoter and take a ribosome binding site and expect to be able to get some kind of this kind of production level. A number of tools which help us provide that. But we're not yet there at the stage where we can combine multiple engineered strains.
Okay, because we have, I mean, we've got approaches and we've got systems at the moment where, broadly speaking, somebody could design a genetic circuit and, foundries can assemble variations and try and get the strains tested to find an optimum producer. Do you see that sort of going to the community level where somebody could be, I need a microbial consortium that can produce compound X? What combination of individual strains and control strains would be used to achieve that in an optimal fashion?
I would love that. I would love that. I think, one of the engineering principles that has really been at the heart of synthetic biology and has really enabled synthetic biology to advance so quickly is this idea of modularization, breaking things down into parts which we can understand the distinct part and then link them together and have some idea of what's going to happen. So that should be applicable at every level, not just at the strain level.
So fantastic. So I guess let's double check one more time. So we answer all the question in the chat and Q&A. So now it's my time 11:08. So I probably need to close. Even though I have question, I will ask question at the next session. So thank all for joining and staying today again. Although many people are still catching up, accumulated emails over the weekend, like me. Preparing proposals and new semester. We'll meet again next week on January 20, Thursday, the same time. We'll have Dr. Bruce Littman from Arizona State University, also a National Academic member and AAAS fellow, and also Dr. Yale Michaels from University of British Columbia, now Canada. And as usual, the follow-up Zoom chat will occur via the link in the chat box. So see you soon and next week. And thank you so much.
Thank you.
Thank you.
So I already posted the Zoom link. So if you click that one, this one will ask whether you will go to another Zoom, and then you say yes, and then you will move from this room to another room.
Hopefully I'll see you there.
Okay, and then Ross, I mean, if you have a little bit more time, you could join us, or if you need to catch the train.
Unfortunately, I have to say my goodbyes now, but it's been an absolute pleasure to speak to everyone and to hear a wonderful talk. Thank you.
Yeah, thank you so much. Your insightful, direction. We also need to follow that interesting,
the endless questions I posed with no answer. But thank
Sure.
you, everyone.
Well, very useful questions.
Thank you.
Thanks, Ross. Bye-bye.
So Tony, thank you so much. We'll meet again next week, and then again I want to see you in person somewhere.
All right. Have a great rest of the day and see you next week.
Yep. Thank you so much.
Bye-bye now.
Bye-bye.